Changes in genes like those that cause shortness in pea plants are called genetic mutations. Genetic mutations occur when a cell's DNA instructions are changed, either as a result of external events, such as exposure to chemicals or radiation, or by mistakes made in copying DNA during cell reproduction. To understand how genetic mutations occur, it is first necessary to know how instructions are stored by DNA molecules. To store information, DNA employs a genetic code forming words and sentences from chemicals to create a fantastic language of life that is employed by every living creature. The genetic code language is very similar to written languages that use an alphabet, but with these important differences. First, the genetic code alphabet is made up of only 4 different letters, A, C, T, and G, representing the 4 chemical subunits of DNA. Second, every word in the genetic code language is only 3 letters long. Third, with the exception of 3 punctuation words, all of the other genetic code words are the names of amino acids, which are the chemical components from which proteins are constructed. Fourth, a genetic code sentence of instructions is the same as a gene. And fifth, the order of genetic words in gene sentences determines the order of amino acids in proteins, and in turn, the specific order of amino acids in proteins is what allows them to perform specific tasks. To further understand these concepts, let us imagine that the gene for the growth factor enzyme, T, in pea plants consists of the following chain of genetic code words on the DNA: AAT, GGG, ATA, TTT, CGC. This is an instruction for 5 different amino acids to be placed in the correct order during the construction of this enzyme. For the T enzyme to function properly, it is absolutely essential that the correct amino acids appear in the correct order in the protein chain of the T enzyme. For if just one amino acid is incorrect, the protein structure will change, and usually the enzyme will no longer function properly, which in this case means that the growth factor subunits will remain uncombined and no growth factor will be produced. Hence, the plant will not grow tall. Incorrect enzymes like this are usually the result of genetic mutations that confuse the DNA instructions for making proteins. For example, the production of this incorrect T enzyme resulted from the change of just a single letter in the DNA gene sentence. In this case, a G was substituted for a T in the 3rd word instructions, so that amino acid 7 was substituted for amino acid 3. As might be expected, this is called substitution mutation. A substitution mutation can occur at the time the DNA is replicated, prior to cell division. This type of mutation is fairly common and may explain how, long ago, the P's recessive gene for shortness was created. However, many other less common types of mutations also exist. For example, deletion mutations occur if parts of the gene are deleted during DNA replication. And insertion mutations can also occur during DNA replication if extra DNA is inserted into a gene. A final class of mutations occur when the chromosomes themselves are damaged or broken, or, as is the case with Down syndrome, an entire extra chromosome is present. If mutations such as we have just seen occur in the DNA of non-germ cells, the result can be cell death, tumors, or uncontrolled cell growth, such as occurs in cancer. These mutations, however, are not passed on to the offspring. But if a mutation occurs in the genes of germ cells in the ovaries or testes, the resulting sex cells will contain the defective gene, and following fertilization, that gene will be replicated over and over again as the cells of the fertilized egg divide. And eventually, every cell of the offspring will contain the defective gene. Then, if the offspring with the defective gene mates with another defective gene carrier, the gene will be present on each of the diploid chromosomes, and the most common results are death or genetic diseases. Sometimes genetic diseases are not very serious, as is the case with shortness in pea plants. But in other cases, they can be very damaging, as with the human genetic diseases, cystic fibrosis and sickle cell anemia.